So today for your Thursday activity, you're going to be completing a worksheet on Punnett squares, going through a lot of examples like the ones we did on our notes so that you're feeling really comfortable with this before taking that quiz tomorrow. And we're not just going to do made up examples this time. We're going to focus on a real case study. The color of fur in jaguars. So melanism refers to how much melanin you make, which is a pigment controlling the color for us of our skin or our eyes, and in jaguars, the color of their hair. And we're going to look at a real example here of melanism in jaguars to do some Punnett squares and make some predictions about offspring. Now, jaguars are a cool example to focus on because they're the only member of the Panthera family found in the New World, which refers to kind of everything west of the Atlantic, so North America and South America. They're an apex predator, which means they're at the very top of the food chain, and they're really well adapted to the rainforest habitat that they live in. Now, the coat pattern that they have that I'm sure you've seen before might not look like good camouflage to us, but for them it works really well. The reason why is that most of their prey are colorblind. So we see the world with all these beautiful colors, but if you're colorblind, everything's in black and white. And if we were to see it in black and white like most of its prey does, it would be really hard to figure out, one, that it's there, and two, which way it's moving or how fast it's moving. So this camouflage pattern is really great for jaguars because of the world that they live in and that they're adapted to. Now, unfortunately, their population is declining because of things like deforestation and habitat fragmentation. So when we cut down forests or start building things where they were originally living, as well as poaching and human-wildlife conflict. So just generally humans deciding that they want to kill a jaguar for its pelt or just because they think that it's a threat to their family or their livelihood, unfortunately. Now, before we get into our examples here, let's review a couple of genetics terms. Remember that DNA is that molecule inside of all your cells made up of base pairs, those nucleotides A and T matched together, and G and C. It wraps up really tightly into that double helix and then wraps up again even more into those big chromosomes, which you inherit from both parents. And a mutation is any change in those base pairs. So any change in those A's, T's, G's, and C's, which will change the instructions. Now, we know that one section of that DNA is called a gene. So if we think of all the DNA like a big recipe book, a gene is like one recipe, one page in that book. And the allele, or a different form of the gene, is like one version or one form of a recipe. So maybe there's one page or one gene for chocolate chip cookies, and the alleles are different versions of those chocolate chip cookies. Maybe we make one version with nuts, and then another version has M&Ms for chocolate chips, and another version has twice as much sugar. It's the same recipe, but different versions of it. Now, we know that there's dominant alleles and recessive alleles. And in a case like this, if there's a dominant allele present, it's always going to dominate that recessive allele. So if you're heterozygous, you're only going to see that dominant allele's phenotype because it's literally dominating the other allele. To be homozygous means that you have two identical copies of that gene, two of the same alleles. So you could have two dominant copies or two recessive ones. And to be heterozygous would mean that you have one of each. Remember, homo means same, so two of the same copies, and hetero means different, two different copies. And then lastly, we've got our genotype, the actual alleles that an organism has, the letters like big B or little b, and the phenotype, the physical expression of the gene. So for example, in this case, the color of the jaguar's coat. Now, in jaguars specifically, there are two color forms or two alleles here. The first is yellow with those black spots, and this is the one that we're familiar with that we saw above and that you've probably seen before. And then you may also be familiar with the black panther or black jaguars, but you probably didn't know that black panthers and black jaguars still have those black dots. It's just everything around them is black. So if you look at this picture, it's not a totally different color. It's just filling in all that yellow fur with black. You can still see the dots if you look closely, and you can still see the pattern. Just overall, the panther or the jaguar ends up being darker. And this black color, or this black coat coloration, is caused by a deletion in that MC1R gene. So if we think back to our mutations unit and our genetic diseases, here we're focused on that section of DNA, And if there's a specific deletion, it's going to cause, rather than that yellow coat with black dots, this all black coat. And that gives us the two different alleles there, yellow with black dots or all black. As a result of this, it causes more color to be expressed, and that dark coat is the dominant allele, which means that if you have one black allele here, you're going to be a black jaguar. In order to have that yellow and black pattern, you need to be homozygous recessive. If you have one or two copies of that big B, that black on black allele, you will have the black phenotype. Now, what we're going to be doing in this worksheet is setting up a bunch of Punnett squares with a bunch of different jaguar parents to make predictions about their offspring. So just like we did in our notes, you're going to look at the parent genotype, and let's say here that for dad, he is, let's say that he's homozygous dominant. So up here on the top, I'd write in my two big B alleles. And then let's say that mom is homozygous recessive. So over here, I'd write in my two little b alleles. And then just like we did on our notes, we're going to go down and across for each one of these so that we can see what all of our potential offspring would be. And in this case, it's an easy one. We're going to end up with 100% heterozygous offspring, big B, little b, which will have that black phenotype. So each of the Punnett squares that you're going to do will look just like this. And then down at the bottom, there's going to be that box to tell me what the possible genotypes and phenotypes of our offspring are.